All right, excellent. Good afternoon, class. This afternoon, we have lecture 20. And this is this is not lecture 20, I guess it's lecture 18. Sorry, it's mislabeled there. But this is a life history and growth of fishes. So we're going to look at different life history strategies in fishes. And so this is today's lecture. So with that, I'll put this onto the projection. So this is the life history and growth lecture, lecture 18. Even though it says 20, it's really 18. So we're going to begin by some simple definitions here. The first is fishes have complex life cycles and indeterminate growth. And what this means is these are the two general traits that are shared by most fishes that also then sets them apart from other vertebrates. And we're going to talk about these in detail, but just generally here as an overview, when I say indeterminate growth, this is defined as the continual increase in length or volume that occurs in fishes throughout their lives. Really, the only time fishes stop growing is when they die. This is called indeterminate growth. And so this is a characteristic that fishes that most vertebrates don't have. Mammals, for instance, such as yourself, you're going to grow to a certain height and you're not going to grow much taller than that after you reach sexual maturity. Fishes, on the other hand, will continue to increase in their length even beyond sexual maturity. So indeterminate growth. And the second major characteristic here of most fishes, not all, but most, is larval growth. So they have a developmental stage where they are larvae. And so this is defined then as a transformative process of emerging from an egg as a partially developed larval. And this larva bears little anatomical, physiological, behavioral, or ecological resemblance to the juvenile or adult stage. In many cases, the larvae don't even look at all like a fish. Okay, now, when I say most, but not all, some fishes have direct development. We talked about this. For instance, the hagfishes emerge from their eggs almost looking like a fully developed miniature adult. Sharks, for instance, have live birth where the juveniles are birthed into the water looking just like miniature adult sharks. But also remember that most of the fishes are in fact teleosts, and that most of these bony fishes, in fact, have a larval stage. So even though these are good examples, when we look at percentage of fishes in the world, a vast majority of them have this larval developmental stage. All right, and so those are two very distinct key features. So when we talk about indeterminate growth, they continue to grow throughout their life, although growth may considerably slow down as the fish ages. And so if we look here at this growth curve of indeterminate growing vertebrate, we can sort of see the slope of this line down here. Juveniles are going to grow a lot more quickly, and then they reach this subadult stage. And then once they reach sexual maturity, for instance, the slope of this line becomes less steep. So it's slowing down the rate of growth. It continues though. Benefits of indeterminate growth or continuing to grow in size throughout your life cycle is this. For instance, bigger females produce more eggs. So the larger you are, the larger your mouth is, the more food you can consume, the more energy you can then acquire, the more energy you can then dedicate to reproduction, and therefore your egg number goes up. And this increase in egg number is called fecundity. So the fecundity of a female fish increases. Bigger females produce more eggs. Also, in the case of males, mostly, but in some cases females, the larger you are, the better you're able to defend your territory. For instance, this could be feeding grounds or forage, but also spawning territory, like especially in some species of fishes that defend their nests, that provide then an area for their offspring to thrive in and feed from. You can defend this area to ensure your next generation has a good chance of survival. Also, we talked about armor on fishes and structures to avoid predation. Obviously, the bigger you are, the harder it is to eat. You know, you're more of a formidable opponent in that regard. And so the bigger you get, the more you can defend yourself. And also, the faster you might be or the larger you are as far as competing with your own species, for instance, things like food or territories. So those are some benefits. This actually leads to some pretty complicated size structures in fishes. So a population, for instance, can contain different sized individuals that function almost like different species. And in doing so, they occupy different, what we call ontogenic niches. And what that means is larval fishes, again, the really early developmental stages here of the indeterminate growth organisms. Oftentimes in fishes, these larval fishes feed exclusively on plankton, and most likely it's zooplankton. So they are carnivores. So at their larval stages and juvenile stages, most fishes are carnivores, even though they might transition to an herbivorous diet later in life. So they actually occupy two different trophic levels, from carnivore to herbivore. Also, larger fishes can prey on entirely different prey items. So the bigger they get, for instance, the availability of other prey species might become available. And so one of the classic examples of this is largemouth bass and bluegill, where early in development, oftentimes bluegill will spawn a little bit earlier than largemouth bass. And so the bluegill might actually prey on larval largemouth bass early in their life. But as they get older, the largemouth bass has a faster growth rate and a larger mouth. And later in life, largemouth bass will then prey on bluegill. And so it makes complicated ecological kind of food networks or food webs, because imagine if you were studying the African savanna and at the earliest stages of life, gazelle could eat baby lions, but then later in life, the lions could eat the gazelle. And so for fishes, this is a lot more complicated because they can switch roles as they grow. And then this figure down here just then shows the comparison there between indeterminate growth of vertebrates and then determinate growth of vertebrates, such as mammals like ourselves, where we can grow pretty good, but once we reach sexual maturity, we pretty much stop growing. All right, so that's the difference here, this post-sexual maturity part of the curve, right? So those are some considerations there with indeterminate growth. Now, when we look at early developmental biology, here are some terms that we should understand. This figure here just shows a very simple life history, for instance, of fishes. An egg down here is defined as an organic vessel. I like to call this a lifeboat, really, where really all it is designed to do is provide some kind of minimal protection where an embryo can then develop. And so the embryo then is really the fertilized organism that lives within this enclosure, right? And embryos are dependent on yolk sac for food, or in the case of sharks, for instance, we talked about oophagy and intrauterine cannibalization. The mother provides some form of sustenance in the viviparous cases. And the egg-laying fishes, again, most fishes lay eggs. The yolk sac provides this. Larvae then are what occur after what hatches out of this egg. And so these are early immature forms of fish prior to what we call metamorphosis. And so we'll talk about metamorphosis here in the next few slides. This is a complex process where, if you look at this, this is a larval fish. But notice these larvae, I mean, they kind of look like fish, but they don't really look like an adult fish, right? In some cases, the juveniles look quite a bit different than the adults. And so this is a pretty significant change in anatomy, if you will, from larval embryonic fishes to the juveniles and then to the adults. So lots of different, you might even think that these are different species, but they actually are all just different life stages of the same species. So the juveniles are then defined as a post-larvae that are not yet sexually mature. And then adults then are the key feature here is that they produce gametes. They are sexually mature. That is the key feature. They are reproductively active. And here's a term here, allometric growth is, for instance, the increase in size of different organs or parts of an organism at different rates. So like, look at this. Look how big the eye is on this juvenile compared to its body length, compared to the size of the eye in this adult. Right. And so sometimes, for instance, the eye might grow larger early in life, and then the rest of the body sort of grows in to fit the size of that eye. And so not every structure necessarily grows at the same rate, but they all eventually get here. Okay. And in some cases, larval fishes or the embryonic ones, look at this, they don't even have fins. So fins don't even exist in larval fish. So lots of complicated changes occur during what we call early development or ontogeny. When we look at fecundity and fertility of fishes, there's lots of variation here. I talked about the mola mola, ocean sunfish, and I poked a few jokes at this beast as being the most derived teleost, but also the most ridiculous one. Most fecund fish, one female can produce hundreds of millions of eggs. So very fecund, what we say. So fecundity is the number of eggs released by a female during a spawning event in the reproductive cycle. And fecundity decreases with increasing egg size. As you might imagine, if you have larger eggs, the female will be able to hold less of them in her body. Likewise, if there's more energy invested in parental care, for instance, regarding a nest or building a nest, nothing is free. It takes energy to do that. And so you might then produce fewer eggs if you dedicate more energy to making or more energy to caring for them. And typically speaking, like I said, fecundity increases with body size of the female, and this is why indeterminate growth is a strategy. Fertility is then defined as the number of viable offsprings that can be produced from the spawn. So here are your two definitions. So mola mola is what we call an artist-selected species. It produces millions and millions of eggs. This thing really doesn't take care of those eggs. It literally spawns them, external fertilization, and the water sort of takes them where they need to go. Its input or investment is in just making a massive amount of eggs. Now, sharks, like this gray nurse shark here, Carcharodontis taurus, has interuterine cannibalism. The first embryo to hatch eats all the other eggs and embryos within the uterus of the female. And then two pups are born, one from each uterus. So it might only produce two offspring. This thing can produce hundreds of millions. But this thing is going to make a big investment to make sure that those two pups have a very good chance of survival. Whereas this one here doesn't really care. It just sort of says, good luck, hope the best to you. And so when we look at this then on generalized egg strategies, oviparous, look at this, viviparous, get out of here. Oviparous is egg laying. Most fishes lay eggs. Most marine species spawn pelagic floating eggs that are fertilized externally and then float away individually somewhere near the surface in what we call the littoral zone or the euphotic zone of the sea, the ocean. So this is a typo, oviparous, not viviparous is live bearers. Most freshwater fishes and some coastal marine species produce what we call demersal eggs. So we've got pelagic floating or demersal sinking. And these eggs are then laid on the bottom. So like, for instance, if you're building a nest, for instance, that you're going to guard, your egg is most likely not designed to float away, because that's pretty hard to guard something that's floating away, right? So you're going to want to guard something that maybe sinks and stays in the same spot. And so whether the eggs float or sink is sort of these are adapted to then whether we're caring for them. For instance, are we spawning them in nests, and are we caring for them? Some eggs are actually adhesive, and we might then design eggs that will stick to some kind of vegetation. And that sort of prevents them from floating away. And so they're what we call adhesive eggs. So lots of different variations here. And when we were going through the different families, I sort of went over a lot of these unique strategies. So oviparous, typo. Now we'll talk about viviparous. This is development of the embryo inside of the body of the parent, eventually leading to some form of live birth. The offspring are provided nutrition by the parent during development. So in the case of oophagy or intrauterine cannibalism, it's literally providing other offspring to eat, as gruesome as that sounds. In some cases, there are trophotania and or other structures that develop between the embryo and the female that are the equivalent of the umbilical cord in mammals. And there's actually a kind of pseudoplacenta, as we call it, that attaches the female to the offspring developing in the uterus. There are also what we call ovoviviparous fishes, and these are embryos that develop inside of some enclosure of an egg. And so they're supplied nutrients from the yolk, but they remain inside of the female's body cavity until they're ready to hatch. So basically, the mother sort of protects them. It's not an egg just floating around that something else can eat. I mean, another fish could potentially eat the mother, but at least she has a potential to swim away and escape, whereas the eggs are sort of, you just sort of hope nothing sees them. And so she sort of provides a little bit of care in the sense that she waits until they're ready to hatch, and then they get birthed out. So these two strategies here require something. It requires internal fertilization, and this is universal among sharks, but it's limited to about a dozen families of bony fishes. This requires some kind of what we call intromittent organ that the males have to inject sperm into the female. And so here are an example of the claspers of a chondrichthyan shark. This is a male. These are inserted in the female to fertilize her eggs. The fallows stethoids have what's called a priapium. Right here, it is involved in fertilization. These are, again, bony fishes. And we talked about the gonopodium. This is a poecilid, the live-bearing poecilids. Here are some cyprinodont fishes. Have this modified anal fin that this is then used for internal fertilization. Now, typically speaking, this is male to female, but we talked about the odd male seahorse, right, that has the pouch, and the female then deposits her eggs in the pouch of the male seahorse, and then the male fertilizes them, and the male retains those fertilized eggs until they hatch, and the male is the one who gives birth, right? Synathidae. So lots of variation here. So these are the live bearers. We talk about reproductive investments. A couple of definitions here. Spermatogenesis, the origin of sperm. Where do they come from? Where are they made? The testis, right? Males make sperm during spermatogenesis. Oogenesis, again, oo is egg, genesis is to create. And the development of eggs occurs within the ovary and progresses through various stages. While the eggs, or really while these oocytes are in the ovary, they're called oocytes. Once they are ovulated, they are then called eggs. In females, there is lots of energy investment. Look at these things. Look at this giant yolk sac, this yellowish bit here. You can see these eyes here, and this little curly thing is the embryo. The rest of this is all yolk. And again, that's not free. Female had to put it in there. Female had to stick that yolk in there. And yolk is mostly protein and fat. So lots of energy goes into eggs. That's why a lot of fishes also specifically prey on the eggs of other fish, because they're very nutritious. So lots of energy investment on the female side of things. Also, there's energy investment. Let's say we're migrating to a spawning area. Well, it's not free. You got to swim there. That takes energy. Also, taking care of your babies. If you've got parental care, that's not free. You got to swim around. You got to chase away predators. You got to police these things to make sure they're protected. That's not free. Energy investment. Males, they have costs like advertisement, competing for mates, for instance, coloration. A lot of tropical fishes have brilliantly colored males and more drab colored females. And guess what? Again, is something as simple as coloration patterns, that's not free. The fish has to physically synthesize those compounds and produce them in the skin to create these colors. That's not free, right? There's a cost to it. Same with displays, like defending territories. Mate competition, nest building. A lot of males are the ones that are responsible for building the nest. That's not free. You got to move around sand or gravel. Catfish have to dig a burrow. The male has to then sit in there just so that some angler can come along and noodle that male out of there, which is taking advantage of the defense of the territory. Like the females, if they have to migrate to a spawning ground, that's not free. They have to swim there. And again, parental care and guarding of the nest. So generally speaking, eggs have a very high energy investment. Sperm, not so much. You can produce billions of sperm, doesn't cost all that much. So the female this is really costly, which is often why females are also the larger of the gender in many cases. We talk here about fertilization. Fertilization occurs when sperm penetrates or is permitted to enter into an egg. Now, unlike mammals who have a chorion, and there's this reaction, this acrosomal reaction, where the head of the sperm is ruptured and this digestive enzyme is released. This sort of helps digest its way into the ooplasm. Fish have what's called a micropyle. And this is a funnel-shaped hole. It's in the membrane. And so what happens is this funnel-shaped hole actually is surrounded by receptors that sort of proteins that sort of are what we call sperm adhesion proteins. And the sperm then are attracted to this hole. And what happens is it's very small. It's actually only big enough to allow a single sperm to enter at once. And once that sperm comes in, it sort of plugs that hole. And so what that does is it prevents polyspermy, or fertilization by more than one sperm. And so fertilization can occur, like this egg, for instance, could be ovulated outside of the body, or it could be retained inside of the body for internal fertilization. And so here's an example, the dwarf perch. These things can actually mate. A female dwarf perch is able to mate with a male, and it can store the sperm in the ovary for 6 to 9 months. And it can ovulate then whenever it wants. So it doesn't need to ovulate and mate at the same time. It can sort of say, well, you know what, I'm going to mate now and just store this sperm until I'm ready to ovulate. And then once those eggs are ovulated, then they'll be fertilized by my stored sperm. The least killifish can store sperm for 10 months. And actually, once they store their sperm, they can actually ovulate 9 different times. They can ovulate once a month, and each one of those ovulation events can be fertilized with that same batch of sperm that they've stored. So it's not necessarily like you have to synchronize male and female. Like the mating doesn't have to necessarily synchronize. It's not like the sperm are ready and the eggs are ready at the same time. That's sort of what we always think about, but not necessarily in fishes. Again, lots of exceptions to the rule here. And so fertilization can be either internal or external, and it can actually be kind of odd like this. Early development. And again, this is tying again together some major concepts. Indirect development. This includes a larval phase with a distinct metamorphosis into the juvenile. And again, the larvae very much oftentimes do not bear any resemblance whatsoever to the adults. And in some cases, the juveniles can be quite odd looking as well. This is in contrast to direct development. The larval stage is either very brief, doesn't occur, is not defined, or literally the offspring hatch out or are released from the female or the egg, looking just like miniature adults. So while the embryo or organism is enclosed within an egg, or in the case of the sharks, the uterus, it is called an embryo. And when the fish hatches from the egg and becomes free swimming, this is called a larvae. And larvae that still have a yolk sac attached to them are called the yolk sac larvae. And I'll show some photos of this. And they're called yolk sac larvae until this yolk is fully absorbed. The yolk nutrients are gone. And then once the yolk is gone, the fish then will switch over to some kind of exogenous feeding. So the yolk is what we call an internal nutrient source. They sort of swim around with this food attached to their belly. And once it's gone, then they use their mouth to go and get their own food. And like I said, most larval fishes, it doesn't matter whether they're an omnivore or an herbivore later in their life, almost all bony fishes are carnivorous, eating zooplankton as their first food sources. They eat tiny aquatic animals. The term fry is used for advanced larvae or early juveniles. So sometimes after the yolk sac is absorbed and they're starting to get things like fins, you can start seeing they look more like a fish. We use the term fry for advanced larvae. So direct development. Larval periods are bypassed or very short-lived in live-bearing fishes and in fishes that possess considerable yolk reserves after hatching. So salmonids, you've got alevins that are a kind of special thing, but like live-bearing fishes, of course, they might undergo larval development, but this occurs inside of the female's body. And really the definition here is if it's free swimming and undergoing this, it's a larvae, right? If it's inside of the female, it's still an embryo, all right? So a little bit of technicality there is really the only difference. So here's a animated GIF of this here with a fish. And so I'll let this pass through a couple times. And so if you look here, this is that egg. This is that egg. You can see the eye developing, and this is that yolk sac then. And you can see the yolk sac is absorbing in, and usually yolk sac absorption then coincides with the formation of the food or with the mouth, because once you, boom, lose the yolk sac, then you got to start getting food on your own. But notice that the mouth is one of the last things that develops because, well, while you got food here, there's no point in getting extra. And so, but notice that like the eye, of course, is one of the first things that develops. And then you can sort of see that here the fins start to form because you got to have locomotion. And oftentimes the caudal fin's the first fin to form, then the dorsal fin. But you can see that like this is a pretty dramatic change that occurs. And really, this is inside the egg. And this right, boom, there's hatch. So you can sort of see that at hatch there, this fish, this salmon, looks very little like an adult at hatch. Once you see the boom right there, the tail coming out, it looks kind of like a snake with a big sac attached to it. And that is a very distinctive, again, when I say this indirect development, this larval stage, I mean, human beings aren't doing this. Fishes do. It's common in their life history. And so with that, here is a video there of zebrafish. And so this is a little bit more under the microscope kind of observation of the embryonic stages. And so what I showed you there is a salmonid undergoing, embryogenesis, a hatch, larvae, and then really larval development into a juvenile. This here is going to be more development of the embryonic zebrafish. And so if you look here, this is the chorion, the egg shell, and this is the embryo itself. This is the yolk here. And these two cells, and developmental biologists, I tell you, these two cells are the embryo, right? And we can tell it's a two-cell embryo because it starts off at fertilization and then it splits into two. And believe it or not, this is called the cleavage stage because it's cleavage. It's these two cells here. And so watch this. You can see how then these cells are going to divide. There's 4, 8, 16, 32, 64, 128, et cetera, until we get to like this here is what we call a blastula. It's a cap of cells, and this is probably time-lapsed only over a few hours. You can see it happens pretty rapidly, and this is when you get a ball of cells here that sort of forms around the yolk sac, and then what happens from this point on is we're going to see then a little bit of reorganization. This blastula is going to start to move. The cells are going to start to move around and organize themselves into layers. And if you remember from developmental biology, it's like mesoderm and ectoderm and all this stuff. And so that's the next step here. And you'll see it then that it starts to spread around the yolk. And see, here it goes. And it's organizing now into tissue layers that are going to then give rise to different things like internal organs versus the skin and things like this. And then one of these ends is going to be the head, one's going to become the tail. So this is where we start talking about where we're going to get, like, which side is the head, this one or this side? And so this is sort of a strip. The embryo sort of forms this strip then that has grown around this ball of yolk. And so one of these sides is going to be the head and the tail. So here we go. Let's put a bet on this. Which side? And you're going to see where the eyes come in. Oh, the somites are coming in here. So this is the segmentation. It's going to give rise to things like the bundles of muscle and things like the vertebra. And so we can start seeing now muscle contractions starting to move around. Oops. And so that is zebrafish development. We'll see what this video here has to show. At NIH.
Researchers who study human development rely on animal models such as zebrafish to gain insight into growth and movement. Zebrafish have an advantage over other animal models because they develop quickly, within just a few days. They are also transparent, enabling researchers to visualize development in real time. A fertilized zebrafish egg has 3 main components: the single cell, the yolk, and a protective membrane called the chorion. After the first cell division, cells continue to divide symmetrically. These steps are controlled by genetic instructions from the mother. But after the 1,000 cell stage, the embryo's genetic instructions take over, a process called the midblastula transition. The next step is called gastrulation, where the embryo forms 3 distinct layers. Genes direct cells in each layer to develop into different types of tissue. Then the eye develops. Later, The embryo moves for the very first time. Pigment cells, which provide color, form in the eyes and trunk of the body. The heart beats and blood cells move through blood vessels. Now the embryo is ready to hatch from the chorion. In 3 more days after the yolk is absorbed, the larvae will begin to eat.
That's pretty cool there. So that is some of the interesting things there about embryos and their development. And again, larval stage indirect development, very unique. And when we say unique, this is what we're talking about. So this is from your reading. So lots of variation among larvae of fishes, and note that a lot of these things don't resemble the adults at all. So this here is a myctophid. So lanternfish. Look at the eyes on these stalks. And look at this, part of the gut, part of the intestines exterior on the outside of the fish. Now you can sort of see it's got like these fin-like structures on it, but the adults don't have the intestine outside. They don't have eyes on stalks. Very unusual. Here's another lanternfish. Again, its intestine on the outside. Here's D. This is a sea bass, is a perciform fish. Same with this one here, C. This is a sea bass larvae. And notice that we've got these, look at this long thing growing out of the dorsal fin. Like, this is odd, right? The adults don't have this. Likewise, look at these giant spikes growing out of here. And I'll tell you, these are defense so that other stuff has a more difficult time eating these things. So this is like an extreme dorsal spine, if you will, and an extreme pelvic fin spine. E here is a cusk eel, family Ophiidae. Look at this crazy thing growing out here out of the pelvic fin. And this here is a squirrelfish, Holocentridae. The only thing that's recognizable here, remember Holocentridae as the characteristically large eye, but look at these spikes. You know, this is basically to say, hey, even though I'm small and defenseless, I'm not very desirable to eat because I got this sharp stuff on me. And so a lot of times these are sharp, kind of spiky things. So Holocentridae, the squirrel fishes. So lots of variation here, and also very little resemblance to what the eventual adults will look like. So with that, I got a video here talking about the importance of fish larvae and also how people study these things. And so here's a pretty cool video talking about that. Because there are people who, by the way, literally just study identification of fish larvae because they're so different from the adults. Just as much as you have to learn the major characteristics of the adult forms, we haven't even really touched on the major characteristics of the juvenile larval forms.
My name is Daniel Altman, and I have some news for you. This year, for the first time, the Larval Fish Conference is taking place in Mallorca, and we organize a workshop to ID fish larvae. Let's take a look.
What is a fish larva? There's no single answer. Ontogeny is not easily divided into segments. And in fact, some invertebrates, so I'll just say that fish do not really have larvae because they don't metamorphose in the same way that so many invertebrates do.
Why to study zooplankton?
It's always the first step to a project. So it starts simple with identifying the species you are interested in. And if you know what the species you are, then you can go up to the bigger questions.
What you're seeing under the microscope is not what's out there in the ocean. The types of characters we're talking about, and you've heard some of this: body shape, developmental milestones, the gut morphology, the swim bladder, where it is, the medial fins and the paired fins. Dorsal and caudal, the medial, the pectoral and the pelvics are the paired. Different fins form in different ways. The timing, the manner of development is taxon specific. And it's about the time that you get the caudal fin to be fully formed that the larvae are picking up meaningful swimming abilities.
Another very important character is the pigmentation pattern. All the fish larvae, they have a variety of pigments, but after the preservation in formalin, only the black pigment remains. Then most of the descriptions are based only on the melanophora, on the black pigment. This allows to identify even at the species level.
And if you already experienced larval identifier, you may want to start looking at the fish larvae under the microscope. That's really what you're here to do. So have fun looking at these guys.
I'm working on the microscope to understand if I'm able to recognize other species of ethioplankton, and I'm start with the shape of the body, and I use these keys. If I find the right shape and the right pigmentation, I write on my book the species.
Everyone kind of has their own method that they follow.
But to me, I always start with counting the myomeres. So that's for me the very important part. That's the first thing I do. I look at the body shape and the myomeres.
Use your eyes and try to put everything together. The Germans have a word for it, Gestalt.
The first thing I do is subdivide everyone into different body shapes. If you can get piles of larvae that share similar body shapes, it's a lot easier because then you can focus your attention on those types of conditions and the species that have that. And then it makes it a lot easier versus just like looking at the whole dish individually.
I think we need more people. We need more students dealing with the larval stages, because then you can really ask important questions, protect species better, if you care about the juveniles and the larvae and not only the adults. So that's to me the crucial point.
And so these types of workshops are important because it passes on some of this knowledge. It exposes the next generation, these early career scientists, to our field, why it's important, why we do it.
And so there is some, like, the study of these things, because they're way different than the adults. And so, even when we talk about taxonomy in this class, most of the notes are focused on the adults because that's what most people are catching. That's what they're eating. That's what they're fishing for. It's not like you have to, if you're going to go fishing for this, you have to have a really, really tiny hook. You're not going fishing for these things. So, I mean, you can go net them and things like that, but you're not catching them, right? So, at least angling. So with that, that's the larval fish. We talk about some really, I guess, examples or exemplar species of looking at, like, studying the differences in life stages. Again, all these different things around this circle is what we call life history or life stages of fishes. The salmon, of course, one of the poster childs. Everyone knows about the Pacific salmon. It's a great story. And so when we look at the process, for instance, of smoltification, this occurs, it's a complex phenomenon that involves changes in young salmon. So eggs hatch into these yolk sac larvae called alevins, and then those turn into these advanced larvae called fry that are still developing their fins and things like that. And then the fry develop some kind of specific pattern of vertical bars here. And these are called par marks because the par will have it. And par are juveniles. These things are living in FW, fresh water, right? And they might be a few months old. And then at some point, they have to develop the ability to move into the ocean to live the rest of their subadulthood into adulthood in the ocean, which is a transition then from fresh water to salt water. And the transition here from a par, which is an old juvenile salmon, to a smolt, which is kind of like a subadult, an adolescent that's ready to migrate into the ocean. This process here is called smoltification. And we talked about osmoregulation of saltwater versus freshwater fishes. And I told you that these processes are entirely different. You're pumping ions and salts in the opposite directions. It's not a simple thing. And really, smoltification importantly involves the pumps in the gill for ions. And so this is a very well-studied phenomenon. It's a very important process, and really it's sort of like lots of developmental biology is conducted on fishes, really, because they're very unique. And so there's lots of science done on this. And the par of the anadromous salmon then spend their juvenile lives in this at sea, or sorry, the par moving to smolt then spend their juvenile lives at sea as smolts, then turn into adults. The adults then reach sexual maturity in the ocean and then return to fresh water to then spawn. So here is this smolt moving from freshwater to saltwater, the adults then moving from saltwater back to freshwater to spawn. And then they spawn in these gravel pits called redds. And I sort of had this, we went over this during the lectures on the salmonids and the families of salmonids. So here's a video then just sort of going over this again. These different checkpoints here are called life history stages. And you can see that they're all very different. You know, this is in freshwater, this is in saltwater. This is a larvae, this is a subadult. They look different. They live in entirely different environments. They have different ecological roles. So very complicated life histories in fishes. So here's another video.
The Atlantic salmon is an anadromous species. This means that the adults live in the ocean and return to freshwater rivers to breed. These salmon can be found in the North Atlantic Ocean and the rivers that flow into it. Atlantic salmon spawn in freshwater in the fall. A female salmon will use its tail to dig a nest in the gravel where her eggs will be deposited. A female lays an average of 7,500 eggs. The gravel nest is called a redd. Over the winter, the eggs develop into small salmon called alevin. Alevin subsist on their attached yolk sac. In May or June, after the yolk sac is depleted, the young salmon, now called fry, emerge from their nest. Only a small percentage of eggs survive to the fry stage. The fry develop into par, with vertical stripes on their sides for camouflage. Juvenile salmon spend 2 or 3 years in freshwater before migrating to the ocean. Before migration, the salmon undergo a physiological transformation called smoltification. This transformation into smolts prepares the salmon for life in a marine habitat. During this process, their gills and organs change, and the fish imprint on the chemical nature of the river. This enables them to find their way back to the place they were born. They migrate to the ocean to feed, grow, and mature. The salmon grow much faster in saltwater than in freshwater. Adults prey on amphipods, krill, and fish like caplin. The salmon spend 1 to 3 years in the ocean, reaching 2 or 3 feet long, before returning to their natal rivers to spawn. They must swim against strong river currents to reach their spawning grounds. They migrate with the help of the Earth's magnetic fields and their sense of smell. Many Atlantic salmon die after the first time they spawn. Very few spawn a second time, and even fewer a third or fourth time. They only live 4 to 6 years. They are preyed on by birds, marine mammals, and large fish. Commercial fishing for wild Atlantic salmon in the United States is prohibited. Only farm-raised Atlantic salmon can be found in the U.S. fish markets. Atlantic salmon farming is controversial because of the many environmental impacts. Chemicals and fish waste from these farms pollute water systems. The farms also rely on unsustainable industrial fishing to provide food for the salmon. Accidents sometimes happen where thousands of farm fish are released into the ocean due to net failure. Even with fishing restrictions, many populations of wild Atlantic salmon are still in decline. Their populations are vulnerable to many threats like pollution and predation by introduced species. Man-made dams and culverts can block access to their quality habitat. For more marine facts, click the subscribe button.
So that is the story of the salmon. It's a historical example. It's one of the first fishes that were really studied, really between the Atlantic salmon and the Pacific salmons. Lots of work has been done on this. We look at, in particular, some of these smoltification changes. Again, here's a par, here's a smolt, loss of this coloration. This is good camouflage in freshwater rivers, but this counter shading, silvery side with a dark, a silvery side with a ventral, a light color with a dorsally dark colored aspect is better camouflage in the open sea. So counter shading versus par barring. The shape, notice that the slimmer and more streamlined here. And the gill function, increased chloride cell number and function, and also then changes in ion permeability in the direction. So remember, a freshwater par is trying to pump salt in, whereas a saltwater smolt is trying to pump salt out, right? And so two different ways the gill operates. And so these changes then have to prepare the fish to anticipate that move from hypoosmotic freshwater into the hyperosmotic saltwater environment. So the gill has to change. And again, as the video indicated, Atlantic salmon may smolt at ages of 1 to 7 years. A lot of times this depends on water temperature and therefore the latitude, right? The further north you go, the colder it is, and so the slower your development, okay? So a lot of times latitude influences that. Another well-developed life history that has been studied is that of the flatfishes. And talk about, if you think smoltification's odd, look at this, right? It's the flounders, the flatfishes. So flatfishes, life begins as a normal bilaterally symmetrical pelagic larvae, right? As odd as larvae are, in this case, the larvae actually probably are the most normal looking thing. The adults are the ones that look odd. They're free swimming pelagic larvae, and then they emerge from eggs and live normal lives, swimming around, eating zooplankton for a month or two, looking just like just looking like a fish larvae. Then metamorphosis begins. Bones are incompletely ossified in the skull, and what happens is the eyes then begin to migrate onto one side of the head. These bones are plastic, they're flexible, they're not ossified, meaning they've not been calcified to be hard bone. The anterior neurocranium, brain, and eye sockets literally physically rotate around, allowing one eye to migrate across the top of the head. Now, I don't know, man. If this was happening to me, I think you'd have a wicked headache. I don't know. This is, it's kind of ridiculous here when you think about it. But this is what they do. The eyes migrate to the other side of the head. And this is well developed or well studied. Things like thyroid hormones and all sorts of hormone cues are what sort of cause this to happen. And so we look at developmental biology. These are well studied for instance, restructuring of bony features, for example. And so the flatfishes are a classical example of studying this. So here's a video then showing time-lapsed photography of this flatfish metamorphosis. So you can sort of see here, the flatfish starts out looking like kind of like a normal larval fish here. Look at this. And then there we go. We start seeing structures like here's our dorsal fin, our median fins, our anal fin. Here's our caudal fin. Look, looks kind of normal. We are eyes on both sides of the head. We're a pelagic larva swimming around eating zooplankton. And then now pay attention to the eyes. in the head. The eyes get larger, looks more, we're getting kind of like a flounder body shape here on the caudal portion of our body. But now we're going to start seeing some major changes here in the head. Look at this. The eye is starting to rotate over. And then now we're sort of moving out. So here's our dorsal fin. Here's our anal fin. Now we're looking more like that flounder shape. And then the eye will continue to migrate over. And there we have it. That is then the final adult sort of form here of what we think of as a flounder. But that's sort of time-lapse photography there showing that process. And say that like talk about looking different than the adult. This is one of those examples where the larva looks way different than the adult, but in fact, the adult looks way different than most fish. So go figure. Also, let me see where we're going to go here next. Because I've got another thing I want to show. This slide here. So I'll go over this slide and I got something else I want to show you. So, when we talk about sexual maturation in adulthood, you know, this is really a development of reproductively functional fish, and it involves three related but pretty different processes, right? And it's almost like in order to do one, you have to do one before you go to the other. So let's start off here by talking about sex determination, and this is the process by which the gender of an individual is decided. And oftentimes this occurs early during ontogeny for most species, but we talked about things like hermaphrodites. A lot of these coral reef fishes, for instance, in tropical areas are hermaphrodites. And so it's not all fishes, but most have a distinct male and a female, and this is oftentimes determined based on genetics. Like in mammals, this is a genetic phenomenon. And in fishes, it also is, but we'll talk about some other ways that this can happen. Second, then, is differentiation. Once this has been determined, differentiation occurs as the development of some kind of recognized gonadal structure. Now, whether that's an ovary with an oocyte contained within it, or a testis that contains sperm, you know, you can observe this. If the gonad has eggs in it or oocytes in it, then you could conclude this is a female. And so this is what we call sexual differentiation. Now, just because they have oocytes and spermata, I wouldn't even say spermatozoa, I'd say spermatogonia or spermatids. They're not mature gametes, because the mature gametes, ovulated eggs, swimming spermatozoa, those are only evident after sexual maturation has occurred. That means that the gametes are ripe and they are ready to produce an offspring. And so this is then what we call sexual maturation. This is the production of what we call viable gametes, the spermatozoa, the swimming sperm, or the ova. The ova are the ovulated eggs. And so this determination here, we have what's called genetic sex determination. Male mammals are what we call heterogametic. The males have an X and a Y gender chromosome, whereas the females have two X gender chromosomes and what we call homogametic. The opposite is true for birds. The female is heterogametic. They are what we call ZW, and the male is what we call homogametic, a ZZ. And back to my master's degree, I used to study sex determination in fishes. Actually, we were looking at muskellunge, and muskellunge are more of a kind of a XY system, but not really. There's something else going on. And so as much as some fishes might have a genetic sex determination mechanism that might underlie sex determination, these chromosomes are the ones that then drive this process, right? The genes are what cause this. And so some fishes have what's called environmental sex determination, where the signals from these genes are modified by environmental cues. Oftentimes, this can be things like climate or temperature. Sometimes it's food availability. In the case of hermaphrodites, like serial hermaphrodites, this could be behavioral, like when the dominant male leaves the reef, the next largest female then becomes a dominant male, or vice versa. When the dominant female leaves, the next largest male takes over and switches into a female. So gender or sex, if you will, is very plastic in fishes. It can switch back and forth. And in some cases, even if there is a known or characterized genetic sex determination system, oftentimes there is a component of the environment that influences this. So for instance, channel catfish, there is a temperature. Water temperature influences the gender of channel catfish. Water temperature influences the gender of flounder. And so here's an article here that I'll show you. I sort of, this article here, when did this came out? This here is Nature Scientific Reports, right? This is a good colleague of mine, a good friend of mine, and a student. I was on this student's advisory committee. She's a PhD student, Jane. And looking at water temperatures and how they masculinize populations of flounder. And this is southern flounder. This study was conducted at North Carolina State University. And it's actually, it's a very interesting study because it shows that as the temperature of the estuary nurseries increases, what this does is it skews the sex ratio of the population. And what happens then is of course, you see a fluctuation in the total biomass of the population, right? Like, you can understand that if all you've got are males in a population, I mean, if there's no females around, what happens to your fishery? It goes away because there's no reproduction. And so again, this idea here, ESD, environmental sex determination influencing the genetic sex determination. And so this article here talks about this. And I just want to point this out because, these are the sampling sites here in North Carolina. And guess who made this map? I mean, I have to do it. You're down here somewhere where is it at? It's in the acknowledgements right here. Your teaching assistant, somewhere along, here we go, acknowledgements. Your teaching assistant here, Linnea Anderson, with assistance in the GIS map. So, helped out with this. So anyhow, I was aware of this study going on. And so that goes to show you that sex determination and environmental cues are very important things in fishes, and they can actually drive this kind of stuff. And again, we understand this for important reasons. Like if we want to predict the patterns of fisheries or population dynamics, it all starts with breeding, and that involves males and females. So that's important, and that's why it's studied. So kind of cool. And also very complicated. And along the way, I talked about serial hermaphrodites versus simultaneous hermaphrodites, and there's lots of variation here, and it's very plastic. So we're going to talk now about death and senescence is sort of the last topic, and then I've got a story. So death. Death means, to die. Senescence is really like to get older, but also to be programmed to die. And so for animals or organisms that reproduce by sexual reproduction, senescence is sort of required in order to maintain diversity, right? So it's sort of terrible to say this, but you have to die because what happens is in order for selection to occur, the adults have to be removed from the population so that they're not competing their entire existence with their offspring. And so pressure can then be applied to that. And that selection pressure then can be used according to all of Darwin's stuff to say the strongest will survive, et cetera, et cetera. So death and senescence go hand in hand. And so in the case of fishes, senescence is something that usually doesn't happen because most fish, to be honest, die by being eaten by something else. Sometimes they accidentally get killed. Sometimes they get an infection and die from some pathogen, a bacteria or virus. Sometimes they do accumulate mutations and get the equivalent of things like cancer, for instance, although like sharks, for instance, don't get cancer. Maybe there's a slow decline in health, and in some kind of they just give up the ghost because their bodies can't keep up with the environment. But most of the time, I would say the number one cause of death in the ocean is being eaten by something else. You die in the stomach of something else. So senescence is really then programmed cell death. This refers to age-related changes that have an adverse effect on an organism and that increase the likelihood of its death. And this is perhaps maybe more common in mammals, less common in teleost fishes. Most fishes die. I'm going to say 99% of fishes die by being eaten by some other predator, whether that's another fish or human beings coming by, fishing them out and eating them. We, in fact, are a predator in our own regard. So the highest mortality of life history stage is, in fact, the eggs and larvae. So if 99% of fish die by being eaten by predators, 99% of eggs and larvae do not survive to adulthood to reproduce. I'm going to say vastly, this is probably closer to 99.99%, to be honest. You could round it up to 100%. It's amazing how these things can survive to begin with. But that's because things like mola mola make 300 million eggs, because 99% or 1% of 300 million is that's still a lot. So that's what, if you think about it like that. So death and senescence come hand in hand. Most fish actually die by being eaten by something else, right? That's the fish eat fish world out there in nature. So when we talk about this, there's age and growth. A lot of times people look at aging fish and to determine things like growth patterns. And so size is generally correlated with age. Older fish are bigger. I mean, that's typically how it goes. Again, they have indeterminate growth, so they continue to grow throughout their lives. Common aging techniques involve counting naturally occurring rings or lines on scales, or otoliths or bones, like a vertebra, just like you would count rings in a tree. So you could age fish using scales, otoliths, ear stones, and vertebrae. Note, these are all relatively hard structures as well, so they preserve relatively well. So, for instance, you can tell, oh, this fish is 1 year old, 2 years old, 3 years old, 4 years old, 4-something years old, and they can measure age by then like the length. A 4-year-old fish, and then measure the length of this fish, right? So here's a video on otoliths talking about how you can age fish to track things like growth patterns. And then I will tell you a story.
Inside the fish's head, there is a bone, or rather an ear stone, which we use to age the fish, and it's more accurately known as an otolith. We use these otoliths to determine the age of the fish in years.
Fish ear bones? Cool.
When I saw like the ear bones, they were just wow. Because I never thought a whale would have an ear bone or a fish.
Just like we have ear bones that help us with balance, orientation, and sound detection, so do fish. In fact, there are 3 pairs of otoliths that lie deep inside a fish's head next to the brain, one of which is too small to see in this X-ray. And as Chris pointed out, they are not true bones, but known as stones as they continue to grow throughout a fish's life. Every year, a unique layer of protein and calcium carbonate is laid down on the otolith's surface, creating rings. And by counting these rings, scientists like Chris can determine a fish's age. So now you might ask, where does Chris get these otoliths?
The otoliths get to our lab from the surveys that we conduct during the summer. We send teams of scientists out onto commercial fishing boats that we contract, and we fish in the Gulf of Alaska and the Bering Sea. We separate all the fish by their species. We weigh them all, we measure them all, and we figure out if they're boy fish or girl fish. After that, we decide which fish do we want to take otoliths from. We take the otoliths out, and and we put them in a collection vial, and then we ship them down to Seattle.
Sometimes Chris can see enough detail on the surface of the otolith to count all of its rings, but usually he cuts them in half with a scalpel blade, and then roasts them in a toaster at 500 degrees for about 5 minutes. Burning the otoliths really brings out the ring so Chris can make the most accurate observation possible.
And so that is aging techniques on fish. Look at how they grow, and then you can correlate those rings then to things like the length of the fish or the weight of it and things like that. And so a lot of times, like, you look at population dynamics of fishes, this is oftentimes what most people do. Because if you catch a fish out of the ocean, it's not like you can figure out how big it was 2 years ago. What you do is you grab fish of different ages, figure out a way to estimate their age, and then create some kind of equation from a database of ages by weights or lengths. So age and growth studies. Pretty cool. So with that, I have a story. Now, I apologize for this. I do. The semester schedule is a little off. And so we started a week early, and typically this lecture always falls the week before Halloween, which is, I guess, coming up here in 10 days. And so this is the frightening, sarcastic fringe-head fish. It's supposed to be the Halloween fish. This is the Halloween lecture, but we're a week early, so I'm sorry about that. So just consider this to be kind of a spooky time, because the sarcastic fringe-head, they have these mouths that are designed to frighten away stuff. So this is the frightening. You learned it here, AEC 441, mouth wrestling with the sarcastic fringe-head. So sarcastic fringe-heads are family Caenopsidae. Right? These are typically found off the Pacific coast of North America. And they're usually, despite this aggressive, like, look at these teeth and things. I mean, these things are kind of terrifying. They're only about 10 inches long. So they're kind of small, but they are fearless and extremely aggressive, and they charge at anything that approaches their burrow. They defend it. And so the sarcastic part of their common name is attributed to their temperament, in the sense that they do not tolerate fools swimming by their areas. Very territorial. So when two fringe heads encounter themselves, they undergo a tremendous territorial battle, because it's hard for them to stand down. And so they will fight. And they do this by distending these mouths like this, and then they mouth wrestle. And they continue to do this until one of the sarcastic fringe heads establishes dominance, in which case then the loser must relocate. It must leave the area. So look at the picture of these things. They're kind of crazy. The Halloween fish, the sarcastic fringe head. So here's a video showing this here.
Producing young is just one challenge. Finding food and somewhere to live are further trials fish must face. The Californian coast. A wide range of species live here. But all this life means competition for living space is intense. Old shells are highly prized. And this one is occupied by a sarcastic fringe head. These fish are exceptionally quarrelsome. They have to be to defend their living space. an octopus. Inadvertently, it's wandered into the fringe head's territory, and that can't be tolerated. The octopus's impressive jab holds the fringe head at bay. There is more to this behavior than being bad-tempered. The fringe head needs to defend its patch if it is to get enough to eat, and the octopus was competition. Crabs are not the easiest of mouthfuls. Because of the shortage of living space, there are constant boundary disputes, especially with other fringe heads. And this one has got too close. Despite the most extravagant threats, neither is prepared to back down. Success, and it's quick to get back to its shell. A fringe head can never drop its guard. There's too much competition.
And that is then your story for today, the sarcastic fringe head. And so that was a former student story. So it gives you an idea there of some of the topics. And so with that, I'm going to stop recording and answer any questions.